I've known him for a few years, but it was just today that I learned that at one point in Montana, and if you get to know him later, he'll probably show you the photo, he saw Sasquatch. So I kid you not, saw a photo, follow up with him, and he's here to talk also about bioinformatics and cool stuff. So take it away, Travis.
The mask hides the smile. I have a photo, find me, I'll share it. Okay, so thanks for the introduction. I'm here to talk about some of the work that folks in my group do. We develop computational tools for sequence annotation. And before I talk about any of those tools, I'm just gonna acknowledge the work of the people who've done almost everything I'm gonna talk about today. Folks on my team on the left, and a couple of collaborators on the right who are involved with everything I'll show, and their pictures will show up again on the relevant slides. So I said we develop computational methods and statistical models for sequence annotation. And what I mean by sequence annotation, whoops, I want to pause that. Maybe I can't. That mouse moves fast. Oh, well, what I mean by sequence annotation is I've got a genome and I'd like to know what's in it, right? And that can involve identification of protein coding genes and the exons that are in those protein coding genes. It can involve identification of other features that you might see, conservation values across the run of the genome, or transposable elements, as has been identified down in the bottom here, either at wide scale, where it shows that in humans, for example, they dominate the genome, and then zooming in to look at particular integration patterns, both integrating intergenically, but also TEs integrating within TEs, and all of the other myriad things that many of us in this room think about when it comes to annotation. And we develop tools that are related to all of those things. And one thing I just want to quickly highlight is that I'm a part of this collaboration that's developed a database of transposable elements called DFAM. And if you're assembling and annotating a genome and you've got transposable elements annotated, or you wish you knew how, you should get in touch with us, because we'd love to absorb those data. We've got TEs for more than 1,000 genomes right now, and it's expanding at scale. But I want to talk, well, we've got a lot of different things. I think this slide's going to show up. Oh, good. A lot of different things going on. I'm not going to talk about most of them. They have to do with speed or accuracy or handling particular oddities that interfere with the ability to annotate things correctly. But I'm going to talk with you specifically about two parts of this, the moving stuff on the left and then the stuff that's on the right, and I'll get into details about those. These are both tools that we've just released that I think will be valuable to folks in this room. So the first of them is a database, I'm sorry, is an open source library for visualizing annotations in the context of biological sequences. So I want to highlight, this is not a genome browser, but it's a JavaScript library that you can use if you're trying to develop a genome browser of your own form with your own particular requirements, or things that are more complicated in some ways. And I'll show you a couple of examples of such increased complications. And the reason this library exists is because we're regularly creating visualizations either to release with our tools or just to debug our stupid tools. And existing libraries didn't do the job. We ended up going straight down to D3. And if you've ever had to build a visual from that, you know it's nice to have a library that can sit on top of it. And so what I'm going to show you are a few examples of that in play. This is just a scalable and movable replacement replica of the transposable element track, the repeat masker track in the UCSC genome browser, which we developed the layout for. And here it is again now in JavaScript. This is a tool that's related still to transposable element annotation. It implements, did it start? Okay, it implements a visualization that we've got for a tool called PolyA, which adjudicates between competing candidate annotations over particular genomic regions. And I'm not going to try to describe everything the tool does, but one key idea here is that we've got some stuff that's sort of genome browser-like, and then a lot of kind of panning and grabbing over particular regions, and demonstration of heat maps that show position-specific confidence values and the sequence alignments that underlie them. And all of those things are produced in a few hundred lines of JavaScript code wrapped around this library, which shows a lot of flexibility in something that's no longer quite a genome browser, maybe useful to some folks in this room. And I want to really demonstrate that you're not building something that's necessarily a genome browser by showing another thing that's involved with the representation of phage integrations in bacterial genomes. The bacterial genomes are circular. We can also represent a linear form of the genome and represent information about the phage integrations and particular subcomponents, the proteins that are part of those. And all of these things are small parts of a very complicated interaction for some software we're releasing called Vibes. And it's, again, a few hundred lines of JavaScript code gets you this complicated interactive environment. And so if you find yourself wanting to visualize stuff that you can't do with a typical genome browser, I would encourage you to take a look at sodaviz.org. Lots of documentation and tutorials that you can use to get familiar with it. The other half, oh good, timing's going well. The other half of what I want to talk about today is the annotation of either pseudogenes or protein coding sequence that suffers from some kind of sequencing error. Okay, an example, just to kind of get you thinking about this, a toy example here of a run of DNA sequence, protein coding sequence. These are codons that of course encode amino acids in triples. And if I have a mutation or a sequencing error that changes one nucleotide, it's a substitution, then of course I change the amino acid that's associated with that position, but it doesn't screw everything else up. But if I have, say, an insertion of a single nucleotide, that will both change that amino acid and it will shove the rest of the codons afterwards out of frame, which if you're actually talking about a functional protein, suddenly makes it no longer a functional protein because you've busted the open reading frame, or maybe it just changes the function, depends. But if you're talking about sequencing error, it interferes with the ability to label or annotate those because sequence alignment of proteins to DNA doesn't account for these sorts of things typically. Deletions, of course, can do the same kind of thing. And so what we're aiming to do is to develop a piece of software that can, with methods that engage in highly sensitive sequence annotation, still account for these kind of frame shift errors in protein coding DNA. What we've done is we've taken the core model that underlies profile hidden Markov model search. This is a tool called Hammer that many people in the room will be familiar with. It's a more sensitive alternative to BLAST, for example. And what I'm showing on the bottom here is just a representation of the dynamic programming matrix that's used for calculating the optimal scoring path that corresponds to a particular alignment. And sometimes this button works and sometimes it doesn't. We've altered that model to allow instead for amino acid to DNA triple, right, alignments, which is no surprise. Those are codons, and it slightly changes the way the dynamic programming matrix works. We can then extend that model to allow for insertions or deletions of one or two nucleotides, which are frame breaking changes, but the model doesn't actually change all that much conceptually, just gets a lot more complicated in terms of algorithm. And with some algorithmic work to try to knock the runtime down and make it feasible at large scale, we end up with a tool called Framer. That's how you pronounce that. It's definitely Framer. I don't care how you thought you were supposed to pronounce it. It's definitely Framer. Tell everybody you know it's Framer. And I want to give you a couple of examples just quickly of Framer in action. Here's one example, backstory that you can ask me about downstream, but there's a particular pseudogenized COB-N gene sitting inside of a bacterial genome. We know it's pseudogenized for complicated reasons, but the underlying problem was that using, say, tBLAST-N, right, which is for translated blast, searching a protein from a nearby genome or the DNA corresponding to it to this particular region, we'd find nothing at all. And if you use profile hidden Markov models, so Hammer in particular, a variant that does translated search, you can find this little brown run in the middle, which is 20-something amino acids of alignment for an almost 1,000 amino acid long protein. And if you use Framer instead, we can get the full length alignment, not quite full length, but nearly full length alignment with both a much larger explanation of what the genomic sequence is, but also an identification in all of these places that are highlighted in kind of a crummy red in this rendering, the places where frameshift mutations are inferred to have occurred. And if we do that over a larger scale of the entire bacterial genome that this thing came from, where there's an expectation of a lot of pseudogenes, again, for complicated reasons, what we see is that with Hammer or T-BLAST-N, the inner part of this circle shows little spotty bits of a lot of these pseudogenized instances of proteins, which happen to be somewhere else in some other genome. And if we take Framer and we apply it to that same genome, we can recover a pretty large fraction of all of those pseudogenes, as you can see here. And so that's an improvement of the annotation. Why does that matter? Oh, if you're doing metagenomics and you've got low abundance sequence where you're likely to have sequencing error, you'd sure like to know what's there despite sequencing error. If you've got eukaryotic sequence where either you have low coverage or there's been pseudogenization, for example, due to polyploidy, you'd sure like to be able to find those, I think, if you want to fully annotate your genome. And so this tool, I think, will give some help doing that. We've got benchmarking studies also that I won't actually get into the details of this, but the big idea is that being higher on this plot is better. And the blue line is Framer, and the other tools are a variety of frameshift aware or not aware translated search tools, all of which don't show the same kind of accuracy. Runtime for this is comparable to NHammer, so for DNA to DNA comparison as well. This is another picture that I'll just skip through that gives the same kind of idea. And the last thing I'll mention is that one last thing I want to say is something that concerns me is maybe this tool will always say there are frame shifts because it's looking for frame shifts. And so we've taken a large benchmark and that consists of sequences that are aligned to each other from the PFAM database of protein domains and check to see, do we call frame shifts when they're not even present in the database? And most of the time the answer is no. Every once in a while we see an example like this where we actually do call frame shifts. All these things in red are bounded frame shifts, which is a little bit disappointing until you look closely at them. And boy, it's hard to see on the screen. But it turns out that in this region, for example, where I'm highlighting here, right, the identity of the frame shifted alignment is quite high, and the corresponding identity in the non-frame shifted, the original alignment, is actually pretty terrible, suggesting that I'll bet there actually was a frame shift that explained the sequence that is observed in one species versus another. So although we have an occasional false frame shift in our benchmark, a lot of those appear to be actual true frame shifts as well. So that's one kind of a sanity check that we've done, and we're investigating this a little bit further. Both of these tools, Soda and Framer, are both available off of my GitHub repo. There's a picture that will get you there. And I think that's a good place for me to stop, just saying we also work on things that are both highly sensitive and fast, and say thanks.